Today, what we're going to talk about in this video is the molecular biology revolution, and this has completely changed how we do genetics in bacteria and all other organisms. One of the cornerstones of this has been DNA sequencing. Hopefully you watched the videos on DNA sequencing that I posted. There were 3 of them. If you haven't watched them, please go watch them now. But what you'll see in this graph is that over the last 3 decades, the amount of bases has gone from around 1 million back in 1980 to hundreds of millions and billions of base pairs. And you can see this is a log scale, and you see this linear increase in the amount of sequence. And you see after about 2000, when we did the human genome, we've started sequencing lots of other genomes. And there's now thousands and thousands of whole genome sequences. There's a huge amount of sequencing data that's been generated. Let's see if you understood those sequencing videos and can compare them and think about what would be best to use. So we're going to have as like this project that we're working on during this lecture, and I'm going to ask you questions about the project as we go through. And our goal is that we've isolated thermophilic bacterium, Bacillus badgeri, that is able to degrade starch. Your research company has directed you to clone the amylase gene, the enzyme that degrades this starch, from this microorganism, and then overexpress it, because you're then going to use that as a product that you sell as a way of generating sugar from starch. First, you decide to sequence the bacterium's entire genome before cloning the gene. You need your results as quickly as possible. Which method would you use? The correct answer is probably a combination of B and C. Sanger sequencing is an older technology. It's slower, and you can't get near as much base pair information. Illumina sequencing and Oxford nanopore both are good methods, but a combination of the two. Illumina sequence gives you very short sequences that are accurate. Oxford nanopore gives you much longer sequences, can be tens and thousands of bases that you can then put together into sequences. That's probably the best method to use. So it's a combination of B and C. So now that you have the sequence, what can you do with it? Well, we talked a little bit about this during the central dogma. And one of the things I want to reemphasize from that is what an open reading frame is. An open reading frame is a sequence on the DNA that has a start codon, has a series of codons, and then has a stop codon. So that's an open reading frame, a putative sequence that should code for a protein. Now, we can figure these out because, number one, we know the rules. We figured out the central dogma, right? So think about a little bit what you do, right? You'd look for start codons, AUGs, and then you look for a series of amino acid codons after that. And there's some other extra helper things that you can figure out. First of all, theoretically, in a random piece of DNA sequence that isn't coding for a protein, there would be a stop codon approximately every 20 amino acids. If you're seeing that there isn't a stop codon after 20 amino acids, you're going to 50 or 100 or 150 amino acids, that's probably an indication that's a real protein. So that should be one high up on your list. The other thing is every organism has a style or codon preference. You can look at that and say, okay, are these codons that this organism normally uses? Or are these rare codons that this organism normally does not use? If a putative open reading frame has a lot of rare codons in it, it's probably not a real protein. If it is using the preferred codons, if an open reading frame is using the preferred codons, it probably is a real open reading frame. So have been programs that incorporate all these rules, and then some other ones that I'm not going to talk about because it's really sophisticated computer programming and algorithm design. But they've been made easily accessible, and one of the easiest things I've found is something called K-base. And this is a thing put out by the USDA that accumulates all sorts of really easy to use programs that you can then apply to a DNA sequence that you have. One of them is Protocol, and this is a program that incorporates all these rules we've just talked about to predict open reading frames. Okay, now you've predicted your open reading frames, and you have all your open reading frames predicted for an amino acid sequence. What can you do with it? You can actually compare those predicted amino acid sequences, those open reading frames, to all the known open reading frames in the world. And a lot of the time we know what those open reading frames do because we've done research on other organisms like E. coli and Bacillus and Staphylococcus, where we already know what the enzymes do. The common program to compare sequences is something I already showed to you. It's called BLAST, and that's for Basic Local Alignment Search Tool. And if you do a BLAST search and you look for a sequence, let's say we took our amylase gene and we compared it, or we took an open reading frame from our Bacillus badgeri, and we then compared it to all the open reading frames of the world, and one of them came up and it had a really good match to an amylase gene. If you look at this sequence here, this hit shows that it's strong red color, and that red color shows where it's matching, and we have a really high score, which means there's a really good match between the amino acid sequence of amylase and our putative open reading frame. This high homology indicates that this protein may have the same function. And this is, for the most part, true. If a protein sequence has a high homology to another protein sequence and the function of that second protein sequence is known, there's a very good probability that your putative sequence performs the same function. You can do this. You can predict the function of all the proteins, all the open reading frames that you found for your whole DNA sequence. And in this, what you're doing is you're assigning open reading frames to pathways. KBase incorporates KEGG, which is the Kyoto Encyclopedia of Genes and Genomes. This is maintained by a group in Japan. And what it has done is it has assembled information on all these different enzymes that organisms have into biochemical pathways. We know all the enzymes that are involved in glycolysis. We know the order that they take. We know all the enzymes that are involved in tryptophan biosynthesis. We know the order of the pathway. If you have an organism that has all the proteins that are involved in tryptophan biosynthesis, you can predict that that organism is probably going to be able to synthesize tryptophan. So you can make very accurate predictions about the capabilities of an organism. What the program does is it assigns each open reading frame to an EC number, which is enzyme commission number. And again, that enzyme commission number tells you what the enzyme putatively probably does. And by doing this, you can ask all sorts of questions about genomes. Here's some examples. So here on the left is Helicobacter pylori, and this is an organism that has been shown to be associated with ulcers. What you can see is there's all sorts of transporters that this microbe has. If you look all around the outside of this, there's tons and tons of transporters. And that actually makes sense because this is a pathogen and it's pulling in lots of things from its host. Also note the urease enzyme up here in the left. This will make ammonia, so it takes urea and converts it into ammonia and increases the pH of the environment around the organism. And this makes sense because its environment is the stomach, and you want to take care of any acid that's there, and it protects the bacteria. Another example is Psychrobacter marinicola. Now, this is an isolate that we isolated last year in Microbiome 551 when we were investigating deep sea ocean vents. We sequenced its genome, and then once we had it, we went through, we predicted all the open reading frames, and then used KEGG to predict, or BLAST and KEGG to predict the function of these things and assemble them into the pathways. And what I'm showing you here is the glycolytic pathway. If you have a blue box, that means there was an open reading frame in Psychrobacter that matched one of these enzymes. So it looks like if you follow this, right, it has the whole glycolytic pathway all the way to pyruvate. And then it has the enzymes that you could make lactate, so it could ferment this to lactic acid. It also looks like it can ferment it to ethanol, but it also looks like it can convert this into acetyl-CoA and then go into the citric acid cycle. So we can make a prediction that 1, this organism can grow on glucose. 2, it probably can ferment to lactic acid and ethanol. And 3, that it also can respire. And then you can test those predictions. So this shows you how powerful this methodology is, where you can take just from a DNA sequence, you can predict all its open reading frames. You can then compare all those open reading frames to the database of all the other proteins that are known, and from that predict pathways and predict functions of the organism. Let's see if this is making sense to you. I'm going to ask you a question. You now have the entire sequence of the genome of Bacillus badgeri. How could you determine the location of the amylase gene? I want you to write out a protocol of what you would do. Okay, so you've written out your protocol. Here is probably what I would do. First, I would predict open reading frames using protocol. Then I would use BLAST to then determine the functions of genes. I would then look for the genes that have similar sequences to known amylases. Right? And then those would be your candidate amylase genes when you're trying to find the amylase gene in the genome. Our next goal is to determine if these candidate amylase genes, because there's probably going to be more than one, see which ones actually have amylase activity. And one way we could do that is to knock out the gene and then see if it still has, if the organism is now not able to grow on starch or degrade starch. And that would indicate that it truly was an amylase gene. One of the ways that you can do this is using transposons or mobile genetic elements. So let's just take a second and explain what those are. Mobile genetic elements are sequences of DNA found in all organisms that are actually capable of moving. They can increase in number in the genome, and they are another form of selfish DNA, besides like viruses and plasmids. Types of mobile genetic elements, there are insertion sequences that only code for functions for moving the DNA, and then what we're interested in, transposons. These have useful genes for the host, such as antibiotic resistance. An example here is shown TN5, a bacterial transposon, and it's actually composed of 2 insertion sequences. And you'll see that you have insertion sequence left and right. The insertion sequence on the right actually has the transposon, and then it has drug resistance in the middle. When a transposon inserts into a genome, it always causes a mutation. This is because mobile genetic elements are 700 to 10,000 base pairs long. In any gene they insert in, they're going to disrupt it. The ends of the transposon have transcription and translation stop sites, and therefore that will stop transcription and translation. Transposons also have selectable markers, such as drug resistance, and that makes it very easy for them to be found in a genome. And then finally, they're safe for humans to use. Other types of mutagenic protocols, such as using UV light or ionizing radiation or mutagenic chemicals, are dangerous to the humans. So transposons are a very convenient way of disrupting genes. So I'm going to talk through an example that we actually use in our advanced microbiology laboratory, and that is PRL27. This has it's a TN5 that's been engineered in a number of different ways. First of all, the transposase has been put outside the insertion sequence. So here's the TN5 with its drug resistance, and the transposase is on a plasmid outside of it. What this does is it allows the transposon to hop only once. And once it's inserted and the plasmid is lost, it will never move again. It also has a hypertransposase that has a very high frequency of transposition, so you get more events. It also has an origin of replication on the transposon, and that allows a number of different things. First of all, this origin is E. coli specific, so when you mate this by conjugation to some other organism, It's a suicide plasmid. The plasmid can't replicate. So the only way the drug resistance survives is if the transposon hops into the chromosome of the target. You can also use this for cloning. And so this transposon inserts into the genome of this organism somewhere. You can then isolate the chromosomal DNA, digest it, ligate it, and then transform the whole mess into E. coli. And this will form a plasmid again, a circle, where the outside flanking regions actually have DNA from the organism. And from this way, you can figure out where the transposon is. Finally, there's an ORET so that you can transfer the plasmid over and conjugate it from one organism to another. So let's go through an experiment and show you how this thing is used. So you will take E. coli, you will mate it with an organism target of interest. In this case, we're using what's called Rhodospirillum rubrum, right? And the plasmid first mates and transfers over to rubrum, and then you put the transposon will then hop into the chromosome, or else there's no transposition. You then select for kanamycin, and that is the drug resistance that's on this. Any rubrum that does not have kanamycin resistance, in other words, doesn't have the transposon inserted into its chromosome, will be lost. And you also add nalidixic acid, which our rubrum in this case is naturally resistant to, and E. coli is not. So all you get on the plate are our rubrum strains that actually have a transposon in them. Once you have this, what we do in the advanced microbiology lab is we will then screen for different mutations. So you will take all the colonies that come up that are Rhodospirillum rubrum, and you will pick them and put them on different plates. You select for the transposon in a rich medium, you plate on a minimal medium, and you look for the absence of growth. That will tell you whether the organism has a mutation such that it can't grow on minimal medium. It's got some nutritional deficiency. You can also look for photopigment mutants. In other words, you just look for different colors. Have you mutated a gene so that the organism can no longer make a certain pigment? Finally, you can look for photosynthetic minus mutants. You can grow under photosynthetic conditions, compare that to a rich plate, and look for organisms that can no longer grow. So there's all sorts of screens that you can do. All right, so now that you understand how you could use a transposon, let's see if you can apply it to our example. We have our Bacillus badgeri, we have some candidate genes, and you want to identify what actually does encode for an amylase degradation gene. The way you're going to do that is you're going to knock it out with the transposon and then see if it can still grow or degrade starch. You expose the strain to a transposon. In this case, we could use our E. coli PRL27 and mate it to Bacillus badgeri. And this transposon contains drug resistance kanamycin. You plate the resulting culture under rich media plus kanamycin and nalidixic acid. It just so happens our Bacillus badgeri is resistant to it. The donor strain E. coli is not. And then you plate it on the rich medium plus kanamycin. Is this a selection or a screen? 12. What must every microbe have that grows on the plate? And finally, you replica plate to a medium that has starch in a rich medium. You then look for microbes that are capable of degrading the starch, and you take the ones that cannot. Is this a selection or a screen? Okay, number 11, this is actually a selection because you're selecting for kanamycin resistance. Number 12, every microbe that grows on this kanamycin plate must contain a transposon. And number 13, this is a screen because what you're looking for is the loss of the ability to degrade starch. Now we're going to talk about CRISPR systems, and what CRISPR stands for is clusters of regularly interspersed short palindromic repeats. There are 3 known systems, type 1, type 2, and type 3. Here I'm showing you an example of a type 2 system that uses the Cas9 protein. The Cas9 protein has been the system that's been of greatest interest. Okay. Cas1 and Cas2 are found in the majority of systems. Cas1 has the endonuclease activity that is necessary for insertion of the system into the genome, and Cas2 seems to have a structural role. This CRISPR system for the microorganism is actually a defense against viruses. So when a virus comes in, if Cas1 and Cas2 can get to it before the virus starts replicating, they will degrade it, they will clip out a identifying region in the virus, and then it will be added to this regular interspersed short palindromic repeats, and this is where they come from. You have this long sequence that has these alternating constant regions, and then these regions in between, shown here in red, that are actually viral DNA. During the processing of this region, you'll get expression of this, and including transcription of this CRISPR region. The endonuclease will split this up into short sequences, and these are these flank consistent regions, and inside you'll have this sequence that is the viral sequence. This will be then added to Cas9, and this protein is in the cytoplasm. In a later infection, if this virus comes in, the homology to the virus will guide the Cas9 system to it, and it will degrade it. So this is a way that once an organism survives the exposure to a virus, any future attempts by this virus to infect the system will be unsuccessful, because the CRISPR system will destroy the virus. Now, scientists started investigating these systems, and what they realized is they could use this CRISPR-Cas9 system to basically modify any piece of DNA that they wanted. They found that there was a guide RNA that you could put in here, and this guide RNA, you needed a conserved region so that it would bind to the CRISPR-Cas9 system, but then you could put in any region that you wanted, and it would guide the CRISPR-Cas9 system to it. So you could mutate any region that you wanted. If you don't provide a substitute piece of homologous DNA that you want to put in, the Cas9 system will come in and it will clip it, and it will break the DNA. And then this will get repaired by the repair systems, but it gets repaired imperfectly, and it creates an indel. So if there's a non-homologous end joining, it creates insertions in the DNA called an indel. This causes a loss in function of that gene. If in eukaryotic systems you provide a homologous piece of DNA with the Cas9 system, the HDR system is homology-directed repair, where a substitute sequence is added and recombined in to fix the repair. So you can direct Cas9 to make this mutation, your donor DNA comes in and repairs the DNA. So that's in eukaryotic systems. In bacteria, you have to provide a repair sequence and a repair enzyme. In most cases, what happens is the red protein, a recombination protein of phagocytic lambda, is used for this purposes. So with this system, you can make mutations in any place in the DNA that you want, just by making a sequence in the guide RNA, or you can change that any DNA sequence any way you want by providing a donor DNA. The beauty of these systems, and what makes CRISPR so powerful, is that it has about a 50% frequency of success. Many of the previous methods that were used for genetic engineering would have much lower frequencies of success, and then you had to have some type of selection to pick out these regions. CRISPR allows you to make mutations and then just manually screen samples until you find the one that you want, and you only have to go through maybe 10 or 15. That means you don't have to have antibiotics or antibiotic genes for these selections, because they're such high frequency events. And literally, it's made it so that we can pretty much cut and paste any genome that we want in any way that we want. Okay, let's go back to our Bacillus badgeri. Given the latest technology that we just talked about, can you think of a better way to test the function of the candidate amylase gene? And of course, the answer is Once you have candidate amylase genes, design guide RNAs, and then use CRISPR systems to make mutations in them and delete them. All right, the last thing I want to talk about is protein expression. What you'll do in protein expressions is you'll clone genes of interest, you'll put them in plasmids, and then you'll overexpress them and make as much protein as you want. One example is the topo cloning systems, and in these systems you have a special overhang of just one base pair, and then you have this topoisomerase enzyme that you attach on the end. Many polymerases used in PCR reactions will cause an overhang of one A base on the end of the PCR product. So you can use that little tiny sticky end to stick to this, and then this topoisomerase will do this reaction and ligate it in. This system is pretty high frequency. You can do these ligations in 5 to 30 minutes. You get lots of PCR products that get cloned in. So basically what you do is you design primers to the region that you're interested in. You then use PCR to amplify that region. You take the PCR fragment that's produced, you run it through this topo cloning system, and it's very quickly cloned into a plasmid. And there's plasmids just to clone the gene. There's plasmids that you can have promoters on it that will overexpress it. So if you use these protein expression systems, there's 4 major systems. There's one in bacteria, in E. coli. Sometimes eukaryotic proteins do not express well. Eukaryotic genes do not express well in E. coli based systems. So there's a number of systems that have been developed in eukaryotes. There's one in yeast, Saccharomyces cerevisiae. There's insect cells, and there's mammalian cells. Here is an example of an E. coli expression vector, and you'll have a promoter. You'll have the Shine-Dalgarno sequence. You'll have these tags, and you can use these tags to purify the protein. And then you'll have the fusion protein in here, and then you'll have a repressor. And the repressor is often the lac repressor and a promoter operator system that you can shut down this promoter here with this repressor, and it prevents unwanted transcription. So normally you'll grow the strain up to a high population, and then you'll put in conditions that relax the repressor, turn on expression from the promoter here, and you get, huge amounts of protein expressed. These tags, an N-terminal, C-terminal tag, these tags can often be used to easily purify the proteins, and makes many of the purification protocols just one-step protocols to purify the protein. I won't go into the details of this, but you're interested, they're called histag proteins, and you can look it up. Okay, finally, you want to clone the gene. So you now have found your amylase gene. What is the most efficient way to do this? How could you make a large amount of this protein? First, what you do is you create primers that flank the region, this amylase gene that you found. You would then run PCR to amplify the fragment, and then you'd use topo cloning to place the fragment into an expression vector. And lastly, you grow up a large amount of this culture and turn on expression of this by relaxing the promoter, relaxing the repressor. We've talked a lot about genetics and genomics, and these technologies have been used to generate all sorts of useful products in modern society. One we talked about before is GMO crops, genetically modified crops. They give a resistance to Roundup. It's also been used in the curing of certain genetic diseases. You can express all sorts of proteins using these systems, polymerases, research substrates, restriction enzymes. There's therapeutics such as insulin, human growth hormone. In detergents, they actually isolate enzymes that way. Now this doesn't use cloning, but lots of enzymes have alkaline phosphatases and proteases in them, and that makes these detergents work better. The sequencing is really revolutionizing things. It will be true soon that you will be able to, as part of your doctor's visit, they will do a blood test, they will sequence your whole genome, and then they will be able to diagnose and say, well, given your genome, you will have more trouble with heart disease. And then there's different modifications you can use in your diet to help with this. There's also been some interesting projects coming forward. One of them was uBiome, which was kind of pop culture nonsense. And in that case, they were determining people's microbiomes and then making a bunch of dubious predictions about what that means. Another one that really isn't nonsense is the American Gut Project, where they're actually, anybody can send in a sample, they will then sequence it and tell you what's in your gut microbiome. And they're using this for comparative studies that are actually legitimate. So there's some stuff that's going on that's nonsense. In fact, uBiome has now filed for bankruptcy. And then there's the American Gut Project, which I think is legitimate and interesting. And of course, lots of this stuff is used as tools in research. The last thing I want to say is CRISPR is a brave new world. There's great potential to treat genetic diseases, diabetes. There's potentials to use it to shut down cancer cells. Might be able to cure HIV. Maybe eliminate Huntington's disease. But there's also great potential for abuse. Any gene can be edited, and a great example are the CRISPR twins in China. In the process of in vitro fertilization, a scientist, which I refuse to name, engineered two twins to make them resistant to HIV by using CRISPR. This is really dangerous because there's a potential for off-site mutations, and in fact, there was recently a paper in Nature that claimed that there were lots of off-site mutations, but then they've had to retract that because of flaws in their design. But again, other people have done other studies that have shown that maybe there really are off-site mutations. This is a new technology. We really need to learn what it's going to do, how many off-site mutations there are, what the risks are before we start applying this to humans. And then there's idiots like biohackers that are actually trying to use CRISPR when they don't really understand what they're doing. One of the developers of this technology, Professor Jennifer Doudna, has called for a public consensus because she can see the dangers of this technology. One of the things that you should think about is, where do you think we should draw the lines? What do you think it's appropriate to use for, and what is it not? In any case, we need to slow down, perfect this technology, and create some rules.